Hi Year 11 and 12 students. Today we're going to be looking at the HL topic of nucleic acids, and we're looking at transcription and gene expression. There'll be a number of videos. I think there's about 3 videos for this PowerPoint. This first video we're just going to be looking at transcription. So might as well begin straight away into transcription. Now, you did look at transcription in the SL content, so it's a bit of a refresher from SL here. I'll put a link as well. This isn't a video to the SL content for transcription. This is actually a SACE Stage 2 biology video on protein synthesis, so transcription and translation. But just a refresher, in transcription, that's when we're having, remember, all the information in our DNA, it's coding for proteins. Okay, in transcription, we're making a genetic copy of that as messenger RNA, and then we take that from the nucleus and send it to the ribosome, where we can create a protein from that information. Now, we also remembering that we have to have the mRNA molecule be synthesized and turned from pre-mRNA into mature mRNA, which goes through RNA splicing. A little bit more complicated for that we'll talk about in the IB curriculum, but this is just, like I said, a little refresher from SL of what transcription actually is. Moving straight into that though, we need to talk about something called the promoter. So in a gene, so a gene codes for a protein, and we make a copy of that gene as messenger RNA in transcription so that we can create a protein from it in translation. Each gene is split up into 3 different parts. There's a promoter, a coding sequence, and a terminator. So the promoter is the region that's found before the coding section of the gene, and it indicates that transcription should begin at this point. Okay, it basically functions as a binding site for RNA polymerase, which is the enzyme responsible for transcription, and gives that binding site so that the RNA polymerase knows to start here. Okay, that's controlled by a variety of different transcription factors, which we'll talk about a little bit later. Transcription factors, though, basically what they'll do is they'll either bind to either proximal control elements, which means they're near the promoter, or they're distal control elements, which are further away from the promoter. Okay, after we've got the promoter, we've also got the coding sequence. So the coding sequence of the gene is the region of DNA that's transcribed by RNA polymerase after the DNA strands have been unwound and separated by RNA polymerase. And that happens when it binds to the promoter, the DNA at that section will unwind and the bonds will be broken. DNA polymerase comes along, makes a copy of that gene until it hits the terminator. Now, when it hits the terminator, the process of transcription is stopped. Okay, the process of termination, is different between prokaryotic and eukaryotic cells. We're only going to focus on eukaryotic cells for this PowerPoint, but just know that it is a different process in prokaryotes and eukaryotes. Before we continue talking about transcription, though, again, we need to learn a few more terms. So in this time, it's the sense and the antisense strand. So with DNA, as we know, it's a double-stranded molecule. Okay, so it's got 2 strands. We've got the sense strand and the antisense strand. So the antisense strand is also referred to as the template strand, and this is the one that the mRNA is going to be built from. So it's acting as the template for the messenger RNA. It's the opposite information than what's actually on the mRNA strand or the RNA strand. Okay, so it's got the complementary bases to the RNA strand. We also have the sense strand, which is referred to as the coding strand. We call that the coding strand because it has the exact same nucleotides, except for uracilthymine, as the messenger RNA molecule will have. Okay, so the template strand, it basically provides the template for the RNA strand, while the coding strand is the genetic information that the RNA strand holds. Okay, in the IB, rather than using coding and template strand, we call the template strand the antisense strand, and we call the coding strand the sense strand. Now, both of these strands of DNA could have a gene that we need to basically create, or we need to synthesize. So it depends on what protein you're looking at, which strand is going to be the sense or anti-strands, and that will be determined based on the gene that we're using to create a protein. Transcription will always happen in the same direction. It always happens from a 5' to a 3' direction, and it's done so in 3 different steps. It's done in initiation, elongation, and termination. Okay. In initiation, we have our RNA polymerase bind to the promoter region. Can't see that in the picture. That will cause the unwinding and the separation of the DNA strands. So that's the initiation process. We then get elongation take place, where RNA polymerase comes along and it binds. It starts to move along the coding sequence, as we can sort of see in this picture, creating mRNA in a 5' to 3' direction. Okay. Once it reaches that termination point of the gene, RNA polymerase will disconnect, as well as the mRNA strand will disconnect, and the DNA will start to wind back up. Okay. In eukaryotic cells, like I said, that's what we're focusing on in this PowerPoint. The RNA strand will then go through a whole lot of modification processes to form mature mRNA, but we'll look at that in another video.